Hello everybody. Today I would like to tell you about high cell density cultivations of Pichia in a microbioreactor system and how we here at VALIDOGEN incorporate it in workflow. The microbioreactor system of our choice is the bioreactor Pro. Now the colleagues from M2P Labs already described it, so I'm not going to go into any details here, just to emphasize that the distinctive feature of the bioreactor Pro for us is the feeding functionality, which enables us to get high cell density fed batch cultivations. Here are some numbers to start with, just to see how the microfermenter cultivations compare to fermentations in a bioreactor. Here we have data for several well-expressed proteins, both in methanol-induced production setup as well as under methanol-free production conditions. In a bioreactor, we usually obtained cell densities in a range of 400 to 600 grams per liter wet cell weight, and in a microfermenter it amounts to 150 to about 300 grams per liter, with multi-gram-per-litre titers in both scales. Now, how does it fit in our strain cultivation workflow? Well, our usual procedure is to perform bioreactor cultivations directly after screening in deeper plates. This transferability works quite well in the vast majority of cases, with an occasional and rare exception. Here's an example of such an exception. We performed a test for secretion of trivalent VHH, where we compared the benchmark strain with a novel productivity improving approach that we've been working on. The benchmark strain produced 1.3 grams per liter of the product in supernatant and grew to more than 500 grams per liter wet cell weight. Now, here we see screening data, and this is relative product titer compared to the benchmark. Benchmark is on the left in orange, and marked with a green arrow is the best producing strain, which is the obvious choice for the bioreactor cultivation. So we carried out the fermentation with the benchmark strain in comparison to this strain number 8, and it didn't go as well as we expected. The strain grew and produced well for a while, but after about 70 to 80 hours of the cultivation, the strain showed reduced methanol tolerance, which eventually resulted in cell lysis and product degradation. Here we always stay on track and keep the task in mind, and our task is to deliver the best producing strain to our client. So here we have two options. Option 1, only if necessary, meaning if there's an adverse effect of the target protein, and it's an overall effect on all strains. In that case, we would apply an adapted fermentation protocol that we developed for such challenging strains. The option 2, the preferred one, is to find and eliminate possible sensitive strains already early in the process. So we went back to the screening results and selected several of the best performing strains for cultivation in the bioreactor. These are the strains marked with green columns, and this is where the bioreactor really proves itself as a valuable addition to our lab. From the microfermentation results, we can immediately identify this strain number 8 that was the best in screening, but performed worse here. So it is the only strain that had a lower relative titer in the microfermentation compared to the screening. All other strains that we cultivated performed even better in a microfermenter. So we selected 5 best performing strains. These are marked with green arrows again for cultivation in 1 liter bioreactor, and obtained very satisfying results. So all strains grew robustly to high cell densities and yielded titers that were multifold from the benchmark strain. So the best performing strain had a more than 4-fold titer improvement from 1.6 to 6.6 grams per liter, and this proves very sound transferability from microfermenter to bioreactor. Now to next case study. Here everything went very smoothly actually between the scales. So it was the test of novel secretion signals. We designated signal 1 and signal 2 in comparison to the most widely used secretion signal from alpha-mating factor from Saccharomyces cerevisiae. Already from the microscale results, we can see that the signal number 1 clearly outperformed the alpha-mating factor. We performed these 12 strains in the bioreactor and obtained very comparable results to the microscale regarding product titer. So the secretion signal 1 was much better than the alpha-mating factor, and all strains showed robust growth to cell densities of about 300 grams per liter. Here we have a direct comparison of relative titer between screening and microfermenter scale. While there were minor shifts in ranking, we can see that the trend stayed the same, with secretion signal number 1 being the most productive one. To get the full picture, we cultivated all 12 strains in the bioreactor, and again the outcome was confirmed. The best performing strains secreted the VHH to a very high titer of 20 grams per liter, and all strains grew well to high cell densities of between 450 and 550 grams per liter wet cell weight. So again, a direct comparison between the 3 scales. Here we see that the transferability worked really well, and we ended up with 4.5 fold titer improvement. To test for a possible bias that's connected to allocation of the individual strains to certain wells in the plate of the bioreactor, we performed 2 separate runs with same strains but on different positions. This was a project for methanol-free production of industrial enzyme that we carried out for a client of ours, where we used co-expressed helper factors as well. So in regarding productivity, we can see in the graph on the right that the best performing strain had 40% higher activity than the parent strain. So like I said, in the second run, we had the well positions reshuffled, and the results were very comparable. So there were no negative surprises. That was the most important thing. There were minor differences in the optical density between wells, but nothing of crucial importance. And concerning the relative activity, the picture stayed basically the same from the first run to the second, where the most productive strains remained on top. The last thing that I would like to show you is that there is some process development work that's possible in microfermenter as well. Here we performed a design of experiment with the strain producing ambivalent VHH. The starting point was a strain that secreted VHH to a titer of 5.6 grams per liter in a bioreactor. We performed a full factorial design with 4 factors and 2 levels, which gives us 16 microfermentations, which we performed in duplicates. The 4 factors that we used were the amount of the nitrogen source, pH value, feed compositions, and feed value. Now of the interactions that we observed, the most interesting one was that the amount of the nitrogen source had a slight positive effect on the product titer at high pH, but negative effect at low pH values. Here we see an overview of the results of all 16 sets of conditions. So these are the mean values of cultivations in duplicates. Marked red are low values, and marked green are high values. As it usually happens in design of experiment setups, some combinations of conditions yielded better results, some worse, but here it is clear that the condition set number 16 outperformed all the other ones, and it had about 30% higher titer compared to standard conditions, which were in the run number 1. So to test this outcome, we performed bioreactor cultivations under standard and optimized conditions and obtained very comparable results to those from the microfermenter, with titer improvement between 30 and 40%. So to sum up, we can say that microfermentations combine the best of both worlds of microscale cultivations, that being their high throughput and the advantages of bioreactor cultivation, such as continuous feeding and tightly controlled conditions, which lead to high cell densities. Microfermentations are very predictive of bioreactor cultivations and are suitable both for methanol-induced and methanol-free production. Achievable are multi-gram-per-litre protein titers, and we can apply the microfermentations within our strain development and our process optimization project as well. which in the end helps us further leverage the benefits of our Unlock Pichia technology platform. Last but not least, I would like to mention that part of the results that were presented here were generated within our partnership with Boehringer Ingelheim, with whom we have a non-exclusive joint technology development cooperation that reaches back to the old days when we were still called VTU technology. That brings me to an end, and thank you very much for your attention.